Guys, grade 12s, finally it is your turn. It is grade 12 Learn Extra Live Life Science. We are carrying on from last week, genetics and genetic engineering, which is so interesting. And Aslam was with us, and I can't wait. How are you, sir?
All the more better seeing you and listening to Belinda on the other side.
I know, this is terrible. I feel so left out when I don't get to go on the show, and especially now. I mean, I missed it with Lou, and now I'm on with you, Aslam. But guys, let us know how you found Ntokozo, one of our top 10 presenters. It was his first show ever, grade 11, and I think he did fantastic. Big up to Macmillan for sponsoring the show. All of our content comes from this awesome Life Sciences for All Macmillan textbook. So now, if you don't know about Random Acts of Kindness, guys, I'm going to tell you quickly what it's about and let you know that we are announcing the winner in this show for our third week of the Random Act of Kindness. What you need to do is you need to go to our events page, pay it forward, post your Random Acts of Kindness on the events wall. So whether it's a picture, whether it's a video, or whether it's just you telling us what you've done to make a difference in someone's life, you could win this awesome Simba hamper. Thanks, Simba, for sponsoring this. Cheetos. And as long as they promise to share with me too. An awesome Mindset t-shirt, a Cheetos little notebook, a lanyard, as well as 5 CDs, guys. That's 5 CDs. So get posting. You still have a chance for your name to go into our little box of tricks, and who knows, we might be calling your name out. So I think, Aslam, let's get the show on the road. This is going to be a super, super fun show. Post your pictures, your questions on facebook.com/Learn Extra, and you can chat to me on Twitter @Learn Extra. So I think, Aslam, after all of that, I'm sorry to take so much time from your show. Let's get the show rocking.
Thank you, Indy, my genie. let's move on. Welcome, Mindsetters. We went through a whole lot of stuff last week regarding genetics. We started with the basics, guys, and I told you last week already that you need to know those basics before we move forward. Whatever we do thereafter is based on what we spoke about: genotype, phenotype, homozygous, heterozygous. Those terms are absolutely crucial for whatever else we're going to do. Now, what are we going to do today? Today we're going to look at how sex is inherited. Now, I see those eyes raising. Don't get too excited. When we discuss that, you'll understand why I say that. We also want to know inheritance of hemophilia and color blindness as sex-linked characteristics. More about that later. We also want to know how would we interpret pedigree diagrams. Further, we look at the importance of genetic engineering. Big word. But in today's time, with the amount of programs you're watching on TV, the CSIs, the medical detectives, all of those shows, NCIS, Law and Order, genetic engineering forms the key to most of the solutions of the crimes. And we spoke about this when we spoke about DNA as well. So we're going to go a little bit further into that. But a little away from the crime scene, more into how genetic engineering helps in medicine, production of medicine, like insulin or vaccines. Also how genetic engineering can help in agriculture. And in agriculture, we even looked at, when we did meiosis, we looked at polyploidy as how that assists in agriculture. Now we're going to look at other avenues of genetic engineering in that field. And just to mention a few, pest-resistant, drought-resistant, improved quality, improved quantity of crop, a larger or a longer shelf life, etc. Then, while we're talking about that, we want to talk about the ethics and the legislation around these things. Around what? Genetic testing, genetic counseling, genetic engineering. We're also going to touch on selective breeding. And we've got to understand today, when we leave here today, when you switch that telly off, or when you change the channel, not now obviously, you must know the difference between selective breeding and genetic engineering. Also look at cloning. And finally, we look at the beliefs, attitudes, and values concerning genetic diseases. That's quite a bit of work to do in one session. Nonetheless, we're going to go through it. What a better way to start, Indy, than a lovely picture.
Is it a boy or a girl?
That's the
What
question.
could it be?
That's the question. So, sorry to disappoint you, you young ones, when I said how sex is determined. When I spoke about how sex is determined, you had a whole lot of other ideas in their mind, indeed.
They were thinking other
They
things.
were thinking heavy
other
stuff.
things.
No, when we say how sex is determined, we talking about how the child that is about to be born, how is it determined for that child to be a boy or a girl? And so there we have the mom there with a nice round tummy, and she wants to know whether she's having a baby boy or a baby girl. And how is this done? Now, remember last week, we went through the whole procedure of P1, genotype, phenotype, etc. And then we went to the last part, and that was the Punnett square. We also spoke about, when we spoke about meiosis, we said that the male has the sex chromosomes XY, and the female has the sex chromosomes XX. And that's what helps us to determine the sex of a baby. So here's the father, XY, these are the gametes. So he can either have a sperm with X gene or a sperm with Y gene, or we call it the X chromosome and the Y chromosome. The mother, on the other hand, does not have this choice. She has only X and another X. So the females can only offer the X chromosome. The males, on the other hand, can either donate or contribute, I would say rather, contribute the X chromosome or the X gene or the Y gene. So if we put this on a Punnett square, that gives you XX, that and that gives you XX again, YX gives you XY or YX. Remember, you can write it either way, XY. So XX is a female and XY is male. So according to this, guys, what are the ratios that we are looking at every time fertilization takes place? What are the probable, note the word, the probable ratios? The probable ratios according to this is obviously 2:2, or rather, made simpler, 1:1. Or we can say 50% is to 50%. That means every time fertilization takes place, according to the laws of probability and according to the laws of genetics, the probability is 50% chance of having a boy child and 50% chance of having a female child. Indeed?
Okay, so what we're actually going to do is we're going to take a tiny quick break, and when we get back, we're going to carry on with the lesson. Please post your questions onto facebook.com/learnextra, and you can chat to me, Indy, on Twitter @learnextra. I'm loving the show. Genetics, life science, it's all about you. See you now after the break. Hi guys, and welcome back, grade 12s. I hope that you have been watching. Who knows? Let me know if you've been watching since grade 10, 11. I would love to know that. So now, if you have this, I'm going to sound like a repeat. This is the most important link you need to know this year, guys. It is learnextra.co.za/live. And what you're going to get at that link is our schedules, our show notes, and as well as our past week's videos. So when you go there, it's like the one-stop shop for everything you're going to need this year. And grade 12s, if you start working now, it's still February. You've still got the rest of the year to do this. And if you join us every day for Learn Extra Live, well, every Wednesday for Learn Extra Live, I promise you are going to do amazingly. Right, Aslam?
That's for
Start
sure.
from the beginning.
That's for sure.
Hey?
All the time. And
Yeah.
they can still go back and watch if they've missed a few lessons.
Exactly.
Good. Let's move on. Welcome back, Mindsetters. Now, the $1 million question, Indy.
Do I get $1 million?
Yes, I don't know. I don't know if I'll afford that.
Okay.
The question
Okay.
is, who contributes? Who determines then? Besides God. Besides God, we're not talking about God now. From the couple, the married couple, husband and wife, who determines whether the child is going to be born is a male or a female? From what I said earlier.
Oh my gosh.
Is that a tough one, Indy?
That is a tough one. I mean, I'll probably tell you like
The
urban myths.
male must have the Y chromosome. So where's the Y chromosome? Only in the father. So only the father determines whether that child is going to be male or female. Again, to all those very religious people, including myself, we're not talking about God in this equation that we're talking about. We're talking about the probability factor. So the male is the contributor. So James, Abraham, John, all those guys in the background.
Not Belinda,
Not
who's our
Belinda.
director.
Just note that if your wife had a girl, another girl, it's not her fault.
It's his fault.
It's his fault because his Y chromosome is too lazy to do the job. Okay? So the male will determine the maleness. It's only the male that contributes the Y chromosome, so therefore it is the male sperm. It depends which sperm reaches the egg. Remember that. And we spoke about the sperms, the different one of the two genes in each of the sperm. Mendel's law of segregation. Each of the two alleles separate into the different sex cells. So that's what we would do. Now remember, in exams, they like to ask this question. Aslam and Indy had 5 children. And the first one was a boy, the second one was a girl, the third one was a girl, the fourth one was a boy, the fifth one is a girl. What is the probability of them having a girl child in the 7th instance? The answer will still be
80%.
50%. Because the probability is still 1:1. Doesn't matter. Just understand this very carefully. When I was in grade 12, Indy, I always thought, not in this particular problem, but I always thought this would be the first child, that's the second child, that's the third child, that's the fourth child in the Punnett square. What this actually means, that every time fertilization takes place, these are the probabilities. It doesn't mean that's number 1, number 2, number 3. You could, and we know for a fact, there are people that have only boy children. There are other couples that have only girl children. So where does this go? This only gives the probability. Remember that. Okay? So remember, whenever you see that question, what is the probability of them getting a boy or a girl? 50-50. It doesn't change, no matter how many children they've had before that. Good. We move on to something a little bit more complicated, but if you listen carefully, and if you use and apply your mind, it should be easy. We're talking about sex-linked traits. Again, don't raise those eyebrows. We are talking about those genes that are on the sex chromosomes. That means a gene that codes for a particular characteristic indeed, which is found on the X or the Y chromosome. Remember, we have 23 sets of chromosomes. 22 of those are autosomes. That means they are normal body chromosomes. And the last pair, pair number 23, are known in a scientific term gonosomes or gonadosomes. That means the sex chromosomes. Those are the ones that determine the sex of the resultant zygote. In addition to this function of determining sex, they may also carry some other characteristics, or rather, they may carry some other genes which code for other characteristics. Those genes that are carried on the sex chromosome are called sex-linked genes. Yup, I nearly fell over the board there, Indy. You didn't see that, luckily.
I would have
saved me.
I would have saved you. I
think you wouldn't have made it from there. Okay. Certain human disorders, like hemophilia, are caused by sex-linked genes. That means the gene that causes hemophilia is carried on the sex chromosome. In this particular case, on the X chromosome. The Y chromosome is much smaller, so therefore chances of it carrying many genes is reduced. It does carry, but much less. And we are more interested in the ones that are carried on the X chromosome. So there we have already explained that whole story there. If a male inherits the recessive allele on an X chromosome from his mother, obviously he will receive the X chromosome from his mother. If we go back, back again. Forward. There we go. Now, remember, here's a male. X, Y. Where did he get the Y from? From the father. That's the Y there, from the father. This X he got from the mother. That's what you've got to keep in your mind as we talk. So if a male inherits the recessive allele on an X chromosome from his mother, he will have hemophilia. If his mother had hemophilia and she transferred, or if she was a carrier even, and she transferred this X chromosome to him, then he would have hemophilia. Why, you ask? Because it's recessive. How come? And I said last week that for the recessive characteristic to show, it must be homozygous. But let's take this case. He has received one X with a hemophilia small h there, superscript, and he has Y. I said earlier that the Y chromosome does not carry the hemophilia gene, so it doesn't even carry the normal gene. So because he doesn't have another gene to counteract this one, even though he has it only one, that means he has it on the X chromosome, he has hemophilia. Whereas a female, if she inherits one from the mother or the father, and the other one is the normal one, she has the normal, the cat is there, so the mice can't play. Remember, when the cat is away, the mice can't play. Even though this is not homozygous, but there is no cat here by the Y. Y can't carry the cat on its back, if you want to put it that way. So therefore, the boy gets hemophilia in that case. We'll look at some examples a little later. If a female inherits the recessive allele, she would not have the disorder as long as the other X chromosome carried normal dominant allele of the gene, as I showed you in the example there. She had the normal one. However, if she had two recessive ones, then obviously there's no cat there, so the mice will play, so she'll be hemophiliac. Hemophilia is just one example. Color blindness, red-green color blindness, is another example of sex-linked disorders. Let's move on. And there we have a table to show what we are talking about. Look at the genotype. XH, capital HY, that means that's the normal gene. So he has normal blood clotting time. Okay, let's just go back again. What is hemophilia? Hemophilia is a condition where the factor that is necessary to cause the blood to clot when you are bleeding, when you have an injury and you get a cut and it starts bleeding, the blood clots. There's a factor that causes that. If that factor is absent, you would be suffering from hemophilia. So the person that has the recessive gene there, in this case here, will have hemophilia. In this case, XX, capital, homozygous, normal. XH, heterozygous female, she's normal, but she carries the recessive allele, so we call her a carrier. She's a carrier. She has the potential of passing this on to her sons especially. Okay? If she has to pass it on to her daughters, then her husband must also carry that gene, so that X small h would make the daughters also hemophiliac. There we have this last scenario there. So these are obviously examples of mutations. Mutation means something going wrong. And here's a classic example. Something's gone wrong. This person cannot clot their blood normally. Other mutations are autosomal. This is a sex-linked one. The others are autosomal, meaning they affect the other chromosomes, the other 22 pairs. And some are recessive, while others are dominant. Okay? We are going to do examples, so don't stress today. I'm trying to push the content and then do questions because we've been falling behind on our question time. So we're going to do questions, and when we do the questions, then the way you answer it and the format will become clearer, okay? So don't stress about that. We're now looking at a pedigree diagram. What is this pedigree diagram that everybody's talking about? It is a chart or a diagram to show graphically the inheritance patterns over a number of generations. Couple of things that we need to know here. Obviously, they are usually used to study the inheritance of genetic disorders, but they can be used for anything. Pedigree diagram can be used to show a simple monohybrid cross as well. A pedigree diagram can be used for anything you want to use it for. It is mainly used by genetic counselors to map out to a potential couple that, look, these are the scenarios in front of you. For example, a particular female has hemophilia, or rather she knows that in her family there was hemophilia. She wants to get married, and the couple are now interested to see whether they're going to have children with hemophilia. So they see a genetic counselor, and he then maps out a pedigree diagram like this and say, look, these are the scenarios that can happen. He cannot tell them, hey, listen, Indy, Aslam, you can't get married because your children may be hemophiliac. All he does is he tells you, look, this is the situation. This is what can happen. After that, Indy and Aslam must decide whether they're going to get married or not.
It's a tough decision.
It's a tough decision.
It's a very tough decision. So what are the risks? What are the percentages? How do we make that decision, Aslam?
This is what we're going to answer when we look at the questions later. And the important thing is, when the counselor gives us this option, and the same with Down syndrome, if you go to a counselor and he wants to know whether you have a risk of having a Down syndrome baby, all he does is he tells you, look, these are the probabilities that you're going to have a baby with Down syndrome. Indy then will have to take the decision, look, this is what Down syndrome is all about. These are the symptoms of Down syndrome. These are the symptoms of hemophilia. These are the symptoms of color blindness. Will I be able to cope with rearing a child like this? So Indy or whoever else will have to make the decision. Okay, and know when you make the decision, any decision, and this is not about life science, this is about life. Any decision you make, you have to take the responsibility of whatever comes after that with the decision.
Wise words.
Okay.
Hey, life lessons as well, guys.
Life lessons all the time. Life science. All right. What we need to know in a pedigree diagram. First of all, you'll notice there are squares and there are circles. There are some that are shaded and there are some that are unshaded. For example, in this particular illustration, we have the circles there, and the key is given to you. Circles are females. Okay? Circles represent females. Squares represent males. So that's the first thing we have to understand. The ones that are shaded usually in a disorder pedigree diagram, the ones that are shaded are the ones that will have the disorder. So we'll ask the questions just now. And the ones that are unshaded are the so-called normal ones or the carriers. They can be carriers also, but normal. Okay? Second thing that we need to know is that there are certain layers. Can you see that there's 1, 2, 3 layers here? These 3 layers represent 3 different generations. This is the first generation, this is the second generation, that is the third generation. Also, we have to note that only when the line interconnects 2 organisms, or 2 individuals rather I should say, and from there another line connects other individuals, like 4, 5, 6, 7. All of these are offspring of 1 and 2. Number 5 is not an offspring of 1 and 2 because it's not connected to 1 and 2. It is only connected to 4. And number 8 is also not their offspring. It is probably the wife of number 7, and this is the husband of number 4, and so on and so on. So a couple of things that you have to remember. Circles, female. Square, male. Shaded, usually the affected organisms or individuals, and unshaded, the normal ones. The different levels, different generations. There must be a connection to be offspring of the one before. And this one here is talking about albinism. That should be an I, albinism. Albinism is not a sex-linked disorder. It is an autosomal disorder. And it is caused by a recessive allele. Now, if we have to complete this particular chart, very simply, remember what I said last week. When the cat is away, the mice will play. Albinism is caused by a recessive disorder. So obviously, if an individual is affected by albinism, it must have homozygous recessive. That is the key in this problem. So what can we do now? We can, and let's say we're using capital A and small a. So we go immediately to the shaded ones and we fill in there small a, 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 small a. So these individuals, number 1, number 8, number 13, number 14, they are all homozygous recessive. Why do we say that? Because albinism is caused by a recessive gene, and a recessive characteristic will only show itself if it is in the homozygous. Remember last week we spoke about a test cross. A test cross is used to determine whether a dominant organism or an organism showing the dominant characteristic is homozygous or heterozygous. For a recessive characteristic, we don't have to do a test because it has to be homozygous. From there, we need to go to the parents now. Let's look at 4, 5, 6, 7. All of them should have a small a. Why? Because the mother had the 2 small a's. So she contributed 1 gene to them, all small a's. So they all have the small a. But are they shaded? No, they're not. So they are normal. So they should have the capital A as well. So they are all heterozygous for this condition. So they will be normal, but they will be carriers. We'll come to number 3 just now. Let's look at 7 and 8. 7 and 8, they have these offspring. Obvious, number 8 would have contributed a small a to number 12. At the same time, because it's normal, it should have the capital letter A to make it normal. So that is how you work that one. Now this is where you get a little confusion or trickiness, but it's not a train smash. All of these offspring are showing the normal condition. And the individual 4 has heterozygous for the condition. Now what about this parent? Because we don't have further evidence, we would then be able to assume that this could be either a or a small a, because we don't have any other suggestions here. And if all these offspring are AA, they could have been AA, or any one of them could have been a small a, because they could have inherited this small gene from the mother and the other normal gene from the father. Now notice, if you do what I just told you, first fill in the recessive characteristics and thereafter work backwards or forwards or upwards or downwards. Guys, I think that has earned you a break.
Fantastic. That's exactly what they're going to do. Okay, guys, you're going to go to a break, and we're going to be back in literally, I think, 2 minutes. Post your questions on the page, facebook.com/learnextra. There are some really, really good questions. Grade 12s, I'm loving the energy, and I hope that you're enjoying the show as much as we are. I'm having such fun. See you now after the break. Hi guys, and welcome back to Learn Extra Live. It is grade 12 life science, and we are having such fun. It's a nice debating topic. Everyone's posting questions on the page, and I think Aslam is going over everything that you are asking. But I will ask him in the last section of the page. I mean, the last section of the show. What am I talking about? I'm just looking at the page here.
Mutation.
I know, mutation. I'm looking on the page here. There is a post from Mochi, and he has posted a picture of him helping his little brother, obviously doing homework. That is so amazing. Paying it forward. Guys, please post your random acts of kindness on the page now, because after the next break, we will be announcing the winner of this awesome prize. Lots of Simba chips. You're going to be eating Simba chips for months. You're going to be wearing this awesome Mindsetter t-shirt, and you're going to win not 1, but 5 CDs. So get posting, find the events, pay it forward, random acts of kindness, and go. But first, I think, Aslam, it's back to you.
Thanks, Indy. Welcome back, guys. We picked up quite a lot of questions about Pedigree. Hendrik says, I'm lost with Pedigree. I'm hoping, Hendrik, from what I've done here now, you are with us. I'm going to go through this very quickly again. What do we do in a Pedigree diagram? We look for the shaded ones, and the shaded ones are usually the affected ones, again, provided that the affected ones are recessive. If that characteristic is not recessive, then it's the reversed. But the recessive one, you will go to whatever the recessive one, even in a normal monohybrid cross, look for the recessive one. When you find the recessive one, you fill in 2 lowercase or small letters into that, whatever letter it may be, small r's or small a's in this case, you fill them all in, as we have done number 13, number 14, number 8, number 1. So you fill in that. And then you go downwards. If there are 2 parents, each parent contributes 1 gene to each of their children. So automatically 4, 5, 6, and 7 get a small a. And because they are not affected and they are not recessive, whichever way you want to look at it, they must have the big letter as well to counteract that recessive characteristic. Hence we have that situation. And that's where you go down. Whatever I've done here, you can do right through. If there's 16 such generations, you can go down 16 generations, working upwards or downwards. Wherever the recessive ones are found first, you work upwards or downwards from there. I'm hoping that clears up any pedigree problems you may have. Lots of kids look at a pedigree diagram and say, oh shucks, how the hell am I going to do this? There's the answer. Look for the recessive, fill in that recessive as 2 small letters wherever the recessive one is, and work backwards and downwards or upwards, whichever way you want to look at it. Hindi?
That's
Does it
fantastic.
work for you?
Yeah, no, it works for me. It works for me. And mindset is, if you still don't understand, please post your questions on the page. And remember, this is a community, so it's not just for us, it's also for you. If you see a question that you understand, you know the answer to, please post your answer, because sometimes by helping other mindsetters out, you're actually teaching yourself at the very same time.
That's for sure. Whatever you teach, hardly ever you're going to forget. Ask me.
So true.
I know that.
Yeah.
Okay. We want to know the difference. I told you at the beginning. We want to know the difference between selective breeding and genetic engineering. And very simple. Selective breeding, English again. Selective breeding. Select, breeding, to mate organisms. So what we're doing? Human beings use different organisms, the best of the organisms obviously, and they select those organisms that have the best quality. If it's horse racing, it would be the horses that run the fastest, that are muscular, they're healthy, they run the fastest. If it is chicken or lamb, it would be the ones that give the best meat, or in the case of chicken, the best eggs. If it's tomatoes or potatoes, it will be the one that gives the largest fruit, tomato. Tomato, by the way, is a fruit in it. It has ovaries. It developed rather from an ovary.
We learned that last week. I think the grade 10.
10 or 11, yes.
The 11s went through that.
Good. So, where the individual chooses these organisms of the same species and mates them, in the hope of getting the best quality. Obviously, you're not going to selectively breed organisms for the worst quality. Who would want to do that? That would be a waste of effort, time, and money. Nobody's going to do that. You want the best from that. So that is selective breeding. So that's the actual, you're not interfering with the genes. You're simply choosing the individuals that show better characteristics and allowing them to interbreed. Whereas genetic engineering involves identifying certain genes, not an individual, but a gene. And how can we get to this? Because of the Genome Project, where they have isolated the different chromosomes and said which gene is found. In other words, they've mapped this whole thing out in our body, our 23 chromosome. They can tell you on chromosome number 20, these are the genes that are found there. These are the genes that code for this and that and the other. We call it the Human Genome Project. So in that way, we can also find out all other defects, and from there we can also develop certain cures for especially genetic disorders. Okay? So they identify those genes and they move them from one organism to another, or of the same species or to a different species, and even they can remove their gene entirely from there. So they are manipulating the genes, genetic engineering, engineering the genes. They are messing with genes. That's what we are talking about. Both obviously are controversial. They have lots of arguments against them, and some will argue for them as well. Just to give you some examples, selective breeding. Here's Brahman cattle. We have good resistance to heat, but poor beef. They're good in a drought, they can survive, but the meat that comes from them, not so nice. And they made those with English shorthorn cattle, they have good beef, but poor heat resistance. So when they mate them, the offspring has the best of both worlds. Therefore, you're getting the Santa Gertrudis cattle, cross of these two breeds. What's the result? It has good meat and it's resistant to heat. So this is how humans can manipulate the organisms to get the best results. And this can be said also with population of maize. What's happening is the seeds for the next generation are chosen from only individuals with the most desirable traits. The others are not used, and therefore we have this process over several generations, and over time the quality of the crop increases. Notice nowhere in this example did they interfere with the genes. They only took the seeds that came from an individual that showed desirable characteristics, and they propagated, that's a law, propagated those seeds, and therefore we're getting over several, it doesn't happen today and tomorrow, over several generations we get only the healthy ones surviving. Don't confuse this with natural selection, which we're going to do in the next lesson. Right? The question is, what is the difference between selective breeding and genetic engineering? And I'm sure from my discussion earlier, you have got that answer. If you have that answer, please post it on, so Indy can give me the thumbs up a little bit later to say that we are getting the idea. That we want to know whether you're understanding what we are saying here. So post your answers there so Indy can give me the thumbs up.
I like that, guys. We want to see 2 thumbs, eh?
What are the advantages of selective breeding? We might get improved organisms. Remember, it's not a guarantee. It's not a guarantee, because we don't know these good organisms that we are breeding, are they homozygous for those conditions or heterozygous? If they're heterozygous, there's going to be problems because they can pass on the so-called bad gene, and that's not going to be good. Okay? They do not need any special tools or lab to do this. They simply put these 2 organisms together. Can be performed easily by farmers and breeders. Undesirable traits from both parents may appear in the offspring. Disease can accumulate. These are, sorry, we missed something there. I should have known that from the time I said undesirable. Can undesirable be an advantage? Indeed?
No,
No,
that's definitely a disadvantage.
Yes, there. So arguments against or disadvantages. Undesirable. Remember I said earlier about heterozygous. If these were 2 heterozygous parents and one of the individual gets that undesirable characteristic, so it can appear in the offspring, and that's not a good thing. Diseases can accumulate in the population. If there's a disease between these organisms, this disease can then accumulate over several generations. They give you an example there. Remember the deaf Dalmatians, boxers with heart disease, labs with hip problems, etc., etc., etc. These are different examples that they are giving you where, because of selective breeding, we basically accentuate certain problems as well. So while there's good, where the good gets better, but the bad gets badder. Worse,
Badder, worst.
Baddest,
Worstest.
worstest. Okay, so that's what can happen. Apart from that, everybody out there would obviously use religion as a disadvantage or an argument against it as well, by saying that we are messing with nature. With Mother Nature. Guys, back to Indy.
Sure, that's an interesting thought, and I think it could be debated for hours and hours. But what we're going to do is we're going to take a quick break, because we are moving into the last 15 minutes of the show. We will be giving away this when we get back. See you now. And welcome back, guys. Mindsetters, we have been looking at all of your questions on the page. There are so much. Aslam has a look through them, and then he kind of picks and chooses, and he figures out what is the most important and what he really needs you guys to know. So trust in Aslam. He is, he's rolling his eyes, but it's true. It really is true. I think he's going to go over some of the questions that he has seen on the page, and then obviously this is happening in the last 2 minutes of the show. Okay, so Aslam, take it away. Which question do we want to start with?
Okay, I'm going to take the question which asks, if 7 and 8 I don't know who
Ah,
asked that
okay,
last
Harry.
question.
It was Harry. Harry has said,
Harry Potter.
yes, Harry Mtembu.
Harry
Hey?
Tembo. Okay.
Okay,
Good.
he says, if 7 and 8 have another child, what is the percentage probability of having a normal child? That's a very good question.
Guys, this is where genetics comes back. This is a pedigree diagram. Do not look at it as something totally different. This is still genetics. He wants to know, Harry wants to know, if 7 and 8 have another child, what is the percentage probability of them having a child who is albino? Albino, yes?
Yeah.
Okay. Now
And
let's
what
look at-
happens if 7, 8, 9?
Eh?
Ah,
Thank you, Indy. Thank
it's
you.
a pleasure.
I was
Any
almost getting
day.
serious with
Any
that question.
day.
But I'm still sharp. I had my vitamins today.
You did.
Okay,
you
so
did.
these are the parents. Who are the parents? The one was heterozygous. The mother was homozygous. Okay? So we're working out across. So this is your P1. The gametes are obviously A. Treat it like any other genetic problem. Here's your gametes. You spread them out. And we're going to say meiosis. I'm going to do all this so that you remember. And thereafter, these are the gametes. And now we have fertilization taking place, and there we draw the square. Whatever is on the left, keep it on the left. A, capital A, small a, 2 small a's there, A. All right. Now what's happening? We want to know the percentage probability. There, A, heterozygous, A, heterozygous, small a, homozygous recessive, small a, homozygous recessive. So what is the percentage probability? 50% chance, or 1. If you're not asking percentage, you're asking what's the ratio? 1:1. You can see it's 2:2, taken down to its simplest, becomes 1:1. So there's a 50% chance of them having an albino child and a 50% chance of having a normal child who would be a carrier. There was another question that I saw as we were running through. Is this pedigree and writing the small letters only for albinism or for other issues as well? Wherever there's a genetic problem, monohybrid problem, if they give it to you in pedigree, whatever the recessive condition is, or color, or shape, that would then get the 2 small letters, lowercase letters, and you work your way around that. I'm not going to waste too much time on that, otherwise we won't get done with what we want to do further. Okay, we're talking about genetic engineering still, and here's one example of genetic engineering, and this is prescribed in a syllabus, so you need to know it. Okay, and this one here is the production of insulin. What do we do here? We remove a string of DNA from bacteria. There we go. There's the string of DNA. It's also called a plasmid. We use a restriction enzyme, which is a chemical scissor, to cut open a space there on that. And then we take a cell from a human, from the pancreas, which the normal gene for insulin is found. We cut that gene off again with restriction enzyme, and we use an enzyme to insert that into that plasmid. And we allow this bacteria to replicate, or rather we can say, because we're talking of this topic, to make clones of itself naturally. Naturally. Why? Because asexual reproduction takes place here. And therefore, every bacterium that develops from this one would be identical to this one. And we then isolate the insulin that has been produced, and we purify it, and we make it ready for use. We keep it in vials. You can then buy it from the pharmacy, and whenever you need to use the insulin to inject yourself, you would then use it. In this way, the insulin is available on demand, advantage. No animal had to be killed for this process, advantage. There is no side effects because it's human insulin, advantage. And we don't have any religious objections. Remember, some people cannot use certain parts of certain animals. So there's 4 advantages. Now let's reverse that. And the question asks you, what's the disadvantage of production of insulin before genetic engineering? Whatever we said for the advantages become disadvantages, but you reverse it. Why? There's religious objections. There are side effects. We have to kill animals to do this. And it's not always available. So guys, can you see, by learning one thing, the advantage of one is the disadvantage of the old, but in reverse. Be very careful. Okay, good. So that was that. And some examples of that. Scientists have engineered chickens to be featherless by removing the gene in chicken DNA that causes them to grow. Remember we said by removing some of the genes. There's an example. Chicken without feathers. Some ladies must be saying, wow, we don't have to do extra work for that.
I'm thinking, shame, that's terrible.
Yes,
Makes my
and
heart very
others,
sad.
like India and I, would be saying shame.
I want to
Good.
cry.
There we have a cabbage now. These cabbage plants are susceptible to being eaten by some bugs. So what they do is they take the gene that produces poison in a scorpion and they insert it into the genes of the cabbage. So the cabbage then becomes bug-proof. The insects won't eat it because it becomes poisonous to them. Fortunately, not for us. Placing antifreeze gene from fish. Taking the antifreeze from fish. Remember, fish live in water. If the water freezes, the fish don't die because they have fluid in their body called antifreeze in inverted commas. So the tomatoes can still grow in cold weather. This one, just an interesting one. Scientists use the bioluminescent gene from a jellyfish to create glowing green mice and other factors that they use for experimentation. Cloning, obviously you would have got that definition by now, is a production of genetically identical individuals from an existing individual. Now this can be done naturally, like in the case of bacteria, etc., or in our lives, we can also make clones of organisms. The most famous of that was Dolly the sheep. Right? You can read more about that. We're not going to go into the detail.
I'll post on the page.
Okay.
Dolly the sheep is posted on the Facebook
there we
page.
Go. Right. Now, the other thing you come across a lot is GM. GM, genetically modified. So you talk about genetically modified organisms. So that is a topic that you need to read. The questions they like to ask there are, what are the arguments for and what are the arguments against these things? That brings us to the questions. And I'm going to go straight to a question that deals with a pedigree first. Somebody said earlier we must do examples of pedigree. Here's a diagram there with hemophilia in a family. There's the story. Now this is hemophilia. Remember, hemophilia, sex-linked. So there they have the story there. Affected, unaffected, and so on. How many family members not affected by hemophilia are carriers? Are carriers in this case. If 4 had to be hemophilia, it had to be X small h y. This H could have come from the mother, well, it had to come from the mother, X small h. Because the father is normal, he'll have the capital H. So that's one. These ones have both, so this is another one. These ones have both, so this is another one. Remember, the father cannot be homozygous or heterozygous because he only has one gene for it. Okay, so there's 3 of them there. Explain why this disorder affects mostly males in the family. Because males do not have the alternate X chromosome with a possibility of a normal gene to counteract the effects of the hemophilia gene. I think with that, I will have to say cheerio for the Cheerios and Indy.
And Aslam, you aren't going anywhere just yet. Guys, we have come to the end of the show. I wish we could do a 2-hour show. I'm having so much fun, and I know that you guys feel the same. But remember, you can watch this on our YouTube channel. Now, I have to be very quick because we are doing the giveaway. But this is the important news, guys. It's not 1 winner today. It is 2 winners today. Yep.
Me and you.
Yay, we won. No, I'm joking. If
Sorry,
only. Okay, Aslam, I'm going to need your help.
Do you trust me, guys? Do you trust me?
I don't know. Hey, some of them are like kind of... Don't look.
There we go.
Come stand right here. Right in front of the camera. Okay. Come. I'm taking up... Okay! This one, and I get so happy. This one, Steffi. Steffi is the winner of our first Random Acts of Kindness giveaway, and she posted...
Don't tell people your dreams.
Show them.
Show them.
But
Wow.
Steffi actually, she posted, I think, about 5 or 6 times. She helped out at a hospital. She looked after her friend. It was incredible. She is incredible. So now, second one. I think we've got about 1 minute left. This is going to be tight. It's always fun.
Awesome!
Indiana! Belinda. Cabello helped painting. He helped his mom paint the entire room last week, Saturday. So well done to these mindsetters.
Well
I'm
done,
so
Cabello and Steffi.
proud. Yeah.
Help yourselves, help others, and help South Africa become a better place.
Yeah, and a big shout out to Lindsay. We love Lindsay works in the background, and you know what, Cabello, Steffi. Hopefully, if you'll come in and fetch the prize in the studio, and then you'll help us, help us help you eat them, because we've all been sitting here looking at all these prizes, and it looks delicious. Thank you so much, all of you mindsetters
You're shouting
who
out
paid
for
it
Lindsay.
forward.
I've got to shout out twice, because without Lindsay, I don't have this.
Exactly. So big up to Lindsay. Thank you so much, grade 12, for an amazing show. See you same time, same place next week. Goodbye.